I think we get started. So good morning, everybody, and welcome to this symposium entitled Touchy-Feely Exposing the Hidden Languages of Our Skin. My name is David Wiley and Miriam Dool. We are the co-chairs for this session, and I'd like to also acknowledge our colleague Vaughan Macefield, who actually played a very important role in putting this symposium together. So this symposium is sponsored by the Physiological Society, and each of you should have on your seat a flyer advertising a call for papers for publication in the Journal of Physiology, which will reflect today's symposium. So I encourage you, if you have manuscripts in preparation, to consider this special issue. It's free to publish in the Journal of Physiology. We have a little advert out here. I'm a former senior editor for the journal. It's free to publish. We accept format-free initial submissions, and we are, I think, globally, internationally recognized as one of the leading physiology journals, but publish across the spectrum of all physiology. So I encourage you to consider making a submission here. So without too much further ado, I'd like to introduce our speakers. We've got 4 speakers this morning. I will chair the first 2 talks from Miriam and Francis. Miriam will switch with me. We'll have questions after each presentation, so speakers have got about 20 minutes, and then we'll have about a 5-minute discussion. This room isn't going to be used until 3 o'clock, I understand, this afternoon, so we've got plenty of time to be able to have a discussion afterwards as well. We can have a little bit of latitude at the end, but we'll go with that. So first of all, introduce my co-chair, Dr. Miriam Dool, whose presentation is entitled The Neurophysiological Profiles of C. nociceptor and C. itch afferents. Miriam.
Okay. Am I supposed to start the presentation or? Oh, okay. Oh, no, okay. There it is. I thought. So thank you very much for this warm welcome, and also, of course, as the co-chair, a warm welcome from me. I'm very excited to be here and to see so many neuroscientists also interested in sensory nerve fiber function, and I'm really honored to be able to talk to you about today about this topic of itch and pain. And my co-chair already introduced me, maybe just a short remark about myself. I'm, as they in Germany now call it, a clinician scientist, so from the clinical side, I'm an MD specializing in internal medicine, working at the University Hospital in Erlangen in Germany. But from my research background, I've always been in neurophysiology, so I've been interested in mechanisms of peripheral nerve fiber dysfunction, for example, in the context of painful diabetic neuropathy or chronic itching as a symptom of various systemic diseases. And I think this is a very good start, already talking about my, okay, doesn't work, sorry. Do I have to point at something as specific with the pointer? Or? Ah, no, it worked. Okay. Maybe just need some time to wake up. So, actually, talking about my research background, it also brings me kind of to my first slide, where I asked, as I guess many of us do now, ChatGPT to create some pictures for my presentation. And I asked to create a person who has either pain or pruritus, which is the medical term for itch. I will use it now, either itch or pruritus as a term. And I guess we can see that this person has a facial expression. He's apparently having an unpleasant sensation. But if we think about these 2 sensations of pain and itch, I guess we can all agree that those are very distinct sensations which we can clearly identify. Either hitting our leg maybe on a chair or getting a mosquito bite are very two different sensations. And maybe then you will wonder if I tell you, or you probably already know, that kind of the same nerve fiber endings of C fibers can signal either pruritic or nociceptive input in the periphery. And probably you remember, all of you remember that from some physiology lectures in the past, that C fibers are the smallest, slowest conducting unmyelinated nerve fibers compared to other nerve fiber classes, especially A fibers, and they have various functions, including signaling noxious heat sensation, pain, but also itch. So I'm just going to shortly introduce you to a method which allows us to further distinguish between C fiber types, and also I think my colleague from Sweden who's going to talk later also uses the method of microneurography. So I hope I don't take anything away from you just now. So this is a method which allows us to directly record C fibers, single action potentials of single C fibers in humans, in healthy individuals, also in patients. And for that, we insert a very thin recording electrode into a peripheral nerve, in our case, the superficial peroneural nerve, usually at the ankle level. And then we put on an electrical stimulation in the receptive fields of this nerve at the dorsum of the foot. And with the stimulation, we can then record action potentials with the with the recording electrode. And this is how usually our recordings look like. So we depict the action potentials of one single C fiber below each other. And so we can follow this throughout several minutes or throughout our protocols. And so the C fibers we are interested in, we can distinguish mainly by their reaction to mechanical stimulation. So here you can see a von Freiherr, which is put onto the skin, and we have a mechanosensitive and mechanoinsensitive C fibers, CMIs or CMs, those are the short forms, and they do not only distinguish by their mechanical reaction, but also how they react to electrical stimulation. CMI fibers have a property: if you stimulate them with more intense or a higher frequency, they will slow down their conduction velocity, and they do it in a more pronounced way than CMs. So for us, this is an established protocol over the last 30 years to distinguish these two main C fiber classes we are interested in the context of pain and itch. And this is a slide provided also by my colleague Barbara Namer, who I've been worked with in Erlangen for several years. She's very proficient in this technique of microneurography. And these two C fiber classes, CM and CMI, do not only differentiate by their properties, but also functionally. So the mechanosensitive fibers are responsible, as you can imagine, for mechanical pain, but they're also implied in non-histaminergic itch. And with that, we think it's kind of an avoidance for the body. So there's an external stimulus which could damage the tissue, and that's kind of the function to avoid this potential harmful stimulus. CMIs, on the other hand, they are also called sleeping nociceptors because they can be sensitized by chemical stimuli, but also more intense heat, and they are implied in histaminergic itch. I'm going to talk about that. And they probably also have a role in pathophysiological states of neuropathic and inflammatory pain. So we think it's more kind of a long-lasting warning if we have CMI input, which tells the body that there's a pathological within the body, and to rest and recover from that. So now you might wonder, I told you we have these C fibers, and they can both kind of react to potential itchy and potential nociceptive input. So how do we now get these really distinct sensations of itch and pain? And there have been several theories over the years. We have evidence for some of them, and maybe it's just a combination of all of them. So I'm just going to shortly introduce them. There is the idea of a labeled line coding, so that we have specific subpopulations of C fibers that can either just react to itchy stimuli or to potential painful stimuli, and this goes all the way up to the central nervous system with a labeled line. It could also be, and I think this was the first idea back in the days, that itch is the little brother of pain, so that a low intensity stimulation in the skin might induce itch, while a higher intensity stimulation might induce pain. There's also the idea that it kind of depends how certain maybe peritogens or algogens reach the nerve fiber endings in the skin. If only few nerve fiber endings are activated, this will result in itch. But if many at the same time activated, this will result in a pain sensation. And we also have newer ideas that also, of course, it matters what kind of fibers are activated, population coding, and also how the fibers then discharge, so atemporal discharge coding. I'm going to show you some data on that later. So talking about itch, you might know if you think about a mosquito bite, itch induced by histamine produces a very distinct phenotype on the skin. You have wheals, you have vasodilatation, you have edema. But on the right side, you can see a picture of a patient. We call it a butterfly sign because the skin is normal where the patient can scratch. So all the things you see, which could be interpreted maybe as something like that, this is only scratch lesions, but actually here the skin is normal. So usually if in chronic itch conditions, the itch is non-histaminergic and the itch often is non-lesional. And with my background in internal medicine, I can tell you that itch is a very relevant symptom for many patients with systemic diseases. Might there be liver diseases, chronic kidney disease, endocrine diseases, or hematological diseases. And all of them are non-histaminergic itch mediated. So, as I already said, and why is that actually important? Is it just a research question? Are we just interested? Of course not. You can imagine that if the itch is non-histaminergic, antihistamines as a first-line treatment, I think that all general practitioners, family doctors use if a patient comes for them with itch, and these patients with chronic itch conditions usually do not respond to antihistamines except the sedative effect, so they get tired and then you don't feel the itch so much, but of course it also affects the quality of life. So this is kind of the clinical background why it's important to understand these mechanisms so we can have more targeted therapeutic strategies for affected patients. So talking about some older data from the 1990s, we know that histaminergic itch probably follows really this labeled line theory I already proposed to you, because Martin Schmelz in Erlangen actually did microneurography recordings of CMI fibers which react to histamine. You see all these jumps to the right, those are all activation periods. And we have a nice kind of time course with the subjective itch rating to histamine application. This was in a healthy volunteer. And you see here on the right that the CM fiber, this mechanosensitive fiber, does a little bit, but not on the other one, does not at all react to histamine, but the CMI fiber here has a lot of activation. And there was further data that, for example, here on the left, they did a peripheral nerve fiber block. And they looked for kind of an area where mechanical stimulation was no longer felt by a healthy individual, but electrical stimulation was. So we think they blocked the CM fibers, but CMI fiber input was still there. And they could still induce, maybe you can see it here, the wheal from histamine and the reddening. So they also concluded that CMI fiber activation is sufficient to cause a histaminergic itch in humans. And there was data for non-human primates that there's also specific spinothalamic tract neurons which are selectively responsive to histamine. So for histaminergic itch, we think it really follows this kind of labeled line. And as I said, coming from internal medicine, I'm not going to go into the much detail of the clinics, but chronic itching is a symptom of, for example, many immune-mediated liver diseases with a prevalence of up to 60 to 70%. And my colleague Andreas Krämer, who I've also worked with in Erlangen, has identified a specific mediator in these patients called lysophosphatidic acid, or shortly LPA. Is this elevated in patients with these liver diseases who have itch compared to those who don't? And if you inject LPA in mice, they will scratch dose dependently. So we continued, and this is what I did a few years ago. We injected LPA in healthy individuals, and what we received was not an itch sensation, but they subjectively reported a dose-dependent burning pain sensation, what was quite curious to us. But if we applied LPA in a different manner, I'm not so sure if you're familiar with cowage. This is a plant which has very small spicules, which can by themselves induce itch, but if you inactivate them by heat, then you can use them to very focally apply substances in the skin. Then we got a mild itch sensation. So it kind of mattered how we applied LPA in the skin. We also did microneurography recordings, and we saw a more intense activation here on the right again of the CMI fibers, which I already introduced to you, and that's what we also saw if we looked over all our fibers that we recorded, that with the spicules we could activate CM fibers strongly, but with the injection we could rather moderately strongly activate CMI fibers. So that's what we kind of concluded, that it matters how nerve fiber endings are reached in the skin, probably a mixture of spatial contrast and population coding for itch and pain for substances which are non-histaminergically mediated. And I already talked about that we also have the idea that how fibers discharge, how they are activated in response to certain stimuli. And this is data also from a PhD student from Barbara Nama that was published in 2023, that what they did, they also did this microneurography recordings, and you can see a CM fiber on the left, CMI on the right, and they applied histamine and they applied beta-alanine, which is a non-histaminergic itch mediator, and you can see activation periods, but you can see that they are clearly different. The histaminergic one is quite chaotic, there's no real order there, but you can see that this activation kind of seems to follow an order. There are pauses, long pauses up to 2.5 minutes in between these activation periods. They have kind of the same frequencies, low frequencies, 20 to 30 hertz, and the same kind of numbers of action potentials. And they termed this kind of a bursting pattern and a slow bursting pattern because of these pauses in between. And they also used other non-histaminergic stimuli and saw that mainly CM fibers were activated and only those had this bursting pattern. And they also applied these substances twice. And this is everything shown here in light red and dark red, that fibers, if you simulate them with the same kind of pruridogen, they react in the same way, either with bursting or non-bursting. So it seems that certain CM fibers have a fiber-specific response pattern. This is just in the end. We also do experiments or testings with patients. And let me just show you shortly in the end. We did kind of a selective C-fiber stimulation with patients who had high-intensity pruritus due to their liver disease. And we saw that if you activate their C-fibers in the skin, they sense more itch. Itch is here shown in red compared to patients who have no or low intensity pruritus who rather sense this sensation as pain, which suggests that there's already a kind of a switch in how C-fiber stimulation is sensed in these patients. This is an ongoing project, just wanted to show it as kind of a last slide that research is going on. So let me just shortly conclude. We saw that there are distinct subtypes of human C-fibers, especially CM and CMI, which contribute differently to itch and pain. We have various coding mechanisms. For histaminergic itch, very likely a labeled line, but for non-histaminergic itch, probably various coding mechanisms play a role. We have this new finding of a slow bursting pattern in CM fibers, and it might also, as we have seen for LPA, vary how mediators reach nerve fiber endings in the skin. And also in patients, we already see kind of altered C-fiber function if they have a chronic itch, which might also contribute to the pathophysiology. And with that, if you now are very interested in itch, because I excited you so much about it, there is a World Congress on Itch taking place in Germany, in Heidelberg. It's a very nice city, as you can see on the picture. Martin Schmelz, also one of the pioneers in itch research, Will is one of the co-organizers. So I cordially invite you to that. And with that, I would like to thank you all for your attention.
Thank you, Miriam. Do you have questions? Any questions? So I'm going to start. I guess my question is the perception of itch versus pain, and so when the signals are integrated and then processed, and then somewhere in the cortex you sense the pain. What is known about the sort of pattern or stimulation that reaches the cortex for itch versus pain?
Yeah, well, that's a great question, actually. Of course, what is not so great that there's often not so much collaboration between researchers working on the peripheral side and the central side. So we, as we know, for pain and also for itch, so there are not that many data on that, especially on patients maybe who have chronic itch. My colleagues have done also a study with fMRI using like peripheral input, potential itchy stimuli, see if there is what kind of areas in the cortex are activated. So probably there is some overlap between itch and pain, but there is no real data which allow us to say that the cortex or the interpretation there is probably the main
even
factor.
taking it one stage back, if you look at this in the DRG or anything like that, can you get any idea in terms of signal processing within, say, the DRG?
Well, also not really. Like I showed you for the histaminergic itch, we clearly have kind of that. We know that there are some receptors and some interneurons important for itch signaling on the DRG level, but it doesn't really tell us enough to say that is clearly the mechanism which allows us to distinguish as a human itch from pain.
Okay. Any other points for Miriam?
Yeah.
Yep. If you want to wait for the mic, just so that everyone can hear, that'd be great. Thank you.
Thank you for such a great talk as well. I was wondering, is there any known mechanisms which cause the transition to chronicity in itch? And we've got theories in pain about potential trauma, physical, psychological trauma, stress-related mechanisms. Is there any mechanism that we think is prompting this transition?
Yeah, it's of course a great question. Actually, I think for itch, there is not like always feels like pain research started a little bit earlier than the research into itch. So we really don't know that much about how chronicity works. Well, we think that one factor could be just the ongoing input or because of, at least for many systemic diseases, we think it's the input of mediators which maybe accumulate during certain diseases, especially for systemic diseases. So it could be the ongoing input into the periphery of a too high amount of potential pruritic mediators, which could then in the long term change how these fibers signal. And this is also kind of what we think we saw with the patients, maybe, that they already have an altered sensation if you stimulate their C fibers. So there is some kind of chronic process already going on which changed that. But what we really don't know if it's on the peripheral level or if it's on the spinal or if it's on the supraspinal level. So there is actually much more research to be done into that.
Okay, yeah, question there.
Great talk. I was just wondering, with the intensity of the stimulus, is there any chance that the pain or itching will turn to pain depending on the intensity? Is there any crosstalk between the two things where one will change into the other?
Yeah, of course, you're completely right. We know that on the spinal level, there are interneurons and there are mechanisms that pain can suppress itch, at least. We know that. And we have experimental evidence, and also we know from clinical experience one can suppress the other, that for example, or can induce the other. For example, opiates as a pain treatment can have itch as a side effect, and we think that's also due to these spinal pathways, and the other way around, pain can also suppress itch pathways, and we have already the data from animal models that support that. And so far, I guess there is no really, we know it, but there's no really clinical impact or no really clinical use we can now draw from that for patients. But yeah, that's definitely the case.
Okay. With that, thank you very much, Miriam, for your presentation, and we'll move on. Thank you. So our second speaker in this session is Professor Francis McGlone from Liverpool John Muir's University.
Not anymore. Manchester Met.
Oh, Manchester Met. Sorry. Apologies.
Story there.
Okay, well, we've got time at the end. So Francis's presentation is entitled The Roles of A beta tactile and C tactile afferents in Discriminative and Affective Touch.
Here's a mouthful.
Francis.
Touchy-feely really sums up what this talk's going to be about, because one's discriminative and one's affective. I'd like to thank Jay Physiol and Carolyn Vaughan for suggesting this. So I'm going to try and unpack these two systems and just describe how the touch has two dimensions to it. We know, I mean, it doesn't need to go into this in terms of how important touch is. You can see some of the narratives here. This particular one I like by Tiffany Field, a remarkable woman who's basically working in the field of touch for maybe 50 odd years, mainly looking at the developmental impact of touch in preterm infants. But I love these sort of cultural differences here that she just noticed observing American teenagers and French teenagers in terms of their physical sort of proximity and touch. But the neurobiological mechanisms that are driving this tactile behaviour have not been really dug into deeply enough. We basically published a paper recently in Frontiers trying to bring touch back in terms of its clinical application. Nobody can touch anybody these days, yeah? Particularly in some cultures, because they criticise for having some sort of scurrilous activity. So touch is really important in social interaction, and we'll try and unpack that. There's a Will Self will pop up in a moment, narrating a book that was written by Isaac Asimov in the mid-1950s, a science fiction writer, and just listen to what he's prophesizing, which I think is already happening.
Fiction novel, The Naked Sun, Isaac Asimov invented a world, Solaria, in which a tiny fixed population of humans live out their days on enormous estates, waited upon by scores of robots. The Solarians' social ambience is something like that of a 19th century Russian novel. A scrupulous attention to social mores and rankings is only enhanced by this bizarre fact. Delivered from material want, or the requirement for sex, all procreation is scientifically managed in birthing centres, a la Brave New World, they have evolved a severe taboo against physical proximity of any kind. Indeed, the Solarians never even occupy the same room together, let alone touch.
We're getting there slowly. This is nothing social about this, but it's translated everything in terms of physical content now to these remote sort of devices. So this is a paper that Charles Spence published a few years ago that basically gives you some idea of the complexity of how, when you're looking at touch, it covers so many different dimensions. This is the bottom line of my talk in terms of what I will hopefully be convincing you of, if you don't know it already, that there is a system of nerve fibres in all mammals, and I think birds as well, basically, that respond to a particular type of touch. Any textbook, even today, probably only has these skin senses described in the textbook. We will add another one, and the neurobiological basis for that fifth sense will be explained. Again, we know a fair amount about the receptors in the skin that are encoding all manner of events that are impinging on the skin surface. A very complex array of receptors, all of which have some functional properties for touch, temperature, itch, as we just described, and these pathways into the central nervous system and beyond. And of course, this field has been lifted enormously in the last few years with Nobel Prizes to these two characters. Actually, only now finding out how that transduction encoding mechanism actually happens for temperature and for touch. So if we look at the skin senses for touch, if you look at the right-hand side, the one we know most about is this myelinated system. And that's the one that everybody knows about. If somebody touches you, if a fly lands on your nose, you feel it immediately. The reason you have that detection is because these myelinated nerves transfer that stimulus to the brain within tens of milliseconds. The other side, of course, is the one that I want to focus mostly on, are these C fibers. You've already heard from Miriam that the C fibers for itch. What they do, they code a more emotional aspects of a physical stimulus rather than the discriminative. And they're traditionally associated with itch and pain. So most textbooks today will just classify these C fibers, afferent C fibers, as nociceptors or proreceptors. We're going to bring another one onto the block here. So this other element, I think, which is important to recognize, anatomy dictates and determines function. If we look at the skin, basically there are two main types. There's glabrous skin, which is the skin on the palms and the soles of the feet, and then the rest of your body is hairy skin. And then a lot of these fast-conducting mechanoreceptors are shared between these two surfaces, but there's one key difference in terms of the nerve fibre I'm going to be unpacking, and that's this C-tactile afferent. Predominantly, this is covering the hairy skin of the body with very limited presentation in the glabrous skin. And again, the glabrous skin is all about exploring the outside world. The hairy skin is yourself and your body. So the fast stuff, we know a fair amount about the receptors that encode for the various aspects of a physical stimulus, rapidly adapting, slowly adapting. And all of these receptors are basically, they're used for handling objects. You know, they come into their epitome with Braille, where blind people can read these little dots of the fluency that you could read with your eyes. And of course, we all know about this homunculus character. There's far more of these receptors in the digits and the lips, and more cortical territory is allocated to them. And so we get this little character here, basically demonstrating the amount of real estate in the brain that's allocated to discriminative touch. The C fibers, of course, of this little CT, has a far more effective role, and again, it doesn't map onto these somatosensory areas, it maps onto areas in the cortex. We've already seen some description of these fiber types. It's just interesting to get your head around exactly how fast these myelinated nerves are in terms of their encoding properties. But the ones I'm going to focus on largely now are these C fibers that Miriam's already described that have very slow conduction velocities. So they're no good for dealing with events that have just happened, yeah, because the information's getting into your brain seconds after the event. So they're not coding things that need a response immediately. They're having some sort of subjective evaluative function. So these C fibers protect. That's one classic function of the C nociceptor, the C proreceptor, and I will describe the C hedonoceptor, for want of a better word, that these evolved in all mammals, and their functional role is one of protection. This CLTM, or this mechanosensitive C fiber, was first discovered by the Swedes, have dominated this whole world, by the way, Ings Ottoman, recording from the saphenous nerve of a cat. He found when he stroked across the receptive field of these units, he got this myelinase activity happening immediately. But then he had the good presence of mind to keep looking at that recording, and here he saw a response to that stroking touch, which came from a C fiber. That was the first description in mammals that these C fibers that responded to touch existed, and he also had the good presence of mind to make sure that it was not a nociceptor. It didn't respond to a tissue-threatening stimulus. And again, the amazing Swedes developed this technique we've already heard about from Miriam of microneurography. It took 50 years before that class of nerve fiber was actually found in humans. And our view is these coelothresher mechanoreceptors are basically the neurobiological substrate for some aspect of touch, which has that reward value to it. So myself and Greg Essick in North Carolina, I think we're the first to actually ask, can we actually quantify an affective property to touch? So we went through a long process building this bloody robot, but it probably cost $500,000. If you're going to talk to a sensory system, you need a stimulator which is as smart as that sensory system you're talking to. So this robot delivers highly tuned velocity and force-controlled stimuli to the skin, and the participant is basically rating on a visual analog scale how pleasant or unpleasant that stimulus is. And what we found is that dependent on the velocity, so these are the three velocities of that stimulus, and this is a visual analog scale logarithmic, is that when we're stroking across the arm at 5 centimeters a second, it's rated as far more pleasant than fast or slow. So we've got a velocity tuning now where people are reliably reporting that this stimulus at 5 is far more pleasant than the fast or slow one. And interestingly enough, when we move that stimulus to the face, to a different body part, and remember this homunculus idea, this is a much more significant, on a log view, response where stroking that same stimulus on the face is rated as far more pleasant than the fast and slow function is still there. So we've got some kind of heterogeneity in the distribution of these nerve fibres across the skin. There's more on the face. Miriam's already talked about this remarkable technique of microneurography. It's transformed all of our insights into somatosensory processes, because we can tap in to a first order neuron and record the activity in response to all of those classes of stimuli that we've seen on touch, temperature, pain, etc. And this is how the C-tactile afferent was discovered, again by Valbo and his colleagues. This video didn't work, by the way, but we've coupled our robot. This is a study done in Gothenburg with Johan Westberg's group. So we're recording some CTs, and we're basically looking at the function of those nerve fibres to different velocities of stroking touch across the receptive field. And what we find, and this is another 2 or 3 years of work, where we're taking the robot, we're stroking across the skin, we're recording some C-tactile afferents, or Lena Loken did most of this work, and look what we're finding in terms of the firing frequency of these CTs, attuned to around about 1 to 3 centimetres per second. This first order neuron is active primarily at velocities that are within this range. The myelinated nerve fibres basically just keep firing their intensity encoding, so the faster you stroke, the more they fire. We then did the psychophysics, so we asked people to rate how pleasant these stimuli were. We get exactly the same inverted U function in terms of this 3 to 5 centimetres velocity stroking is the one that people rate. So this is affective touch, and this basically gave us the basis of our initial pleasant touch hypothesis. And those 3 papers basically describe that story up until 2014. We don't think it's as simple as that. I think these things do more. This neurone paper basically describes all of these different processes in terms of the fast myelinated journey you've already seen up to somatosensory cortex, and then the affective journey, this inverted U function coming into lamina 1. We don't know how it gets from there up to the brain, by the way. We have yet to find out the pathway. But then it maps into areas of paralimbic structures that are basically more interested in encoding emotion. So what's the functional role of these nerve fibres? We call them CLTMRs in animals and CTs in humans. If we look at the kind of activities that we see, again, just summarising that they're not present in glabrous skin. And we're now looking at across species. I want to look at comparative stuff. If you take C. elegans, this little worm, it's got like 340 neurons, I think. If you grow it in a colony, i.e. a lot of touching going on, it grows to its normal healthy length. If you grow it in isolation, where it's getting no touch from conspecifics, it doesn't grow effectively, and its behaviour has changed. So again, we see a developmental aspect here that touch may be important. This is an interesting one. A group of researchers probably had too much ecstasy floating around, so they threw it in the tank where the octopus was, and this octopus behaviour completely changed. It went from the solo, from a sort of, playing on itself, with itself, not that's a bit rude, isn't it? Anyway, playing with toys, and then what the MDMA did, it made that octopus far more social in terms of convening with its other mates. And this is 500 million years of evolution separate these two, but we see serotonin coming into the picture now as a possible role in this affiliative touch. A classic study by Michael Meany, which there are some questions about its veracity, by the way, but nonetheless, I think this is pretty sound, is that pups that are raised from a high-licking grooming mother grow up to be less anxious and far more better behaved and controlled. Those who don't get this licking and grooming in the early days grow up to be stressed and very anxious. And that also affects the way that the brain functions in terms of neuronal structure. So there's something that's clearly happening very early on in life where this tactile input is playing a fundamental role in regulating stress. Again, what's the mechanism? We know a fair amount about endorphins and grooming, another neurotransmitter coming into the picture here. If you look at these grooming monkeys and you block the opiate receptors with naltrexone, they groom less. Sorry, they groom more when you block the opiate receptor. So now we're seeing that this opiate receptor is playing an integral part in the reward associated with close physical contact. You've all heard of Harlow stuff, of course, where this monkey, rather than clinging to where the petrol pump was, or the fuel pump where all the food was, it clings to this useless surrogate, which is just soft. But what Harlow didn't know is that there's a possible mechanism that's describing that behaviour, which is this nerve fibre that promotes the reward of physical contact. And we know that more tragically, the evidence from people like René Spitz, and this crazy man, Ceausescu, who wanted to breed more babies, and these thousands and thousands of infants discovered in these Romanian orphanages, all of which were severely behaviourally damaged by the fact that they, basically the story here is they were fed and watered, but they were not handled. They didn't get any touch. And you look at the significant impact it had on their life. This is another character, Waldhouse Watson, who had these interesting attitudes to how you should bring your child up, which is just quite extraordinary. But when I looked into what his children actually did, we found that they didn't actually fare too well in terms of that rather brutal upbringing where you don't cuddle or touch your children. So this guy is pretty dangerous, actually, so don't listen to his behavior, what he's basically prescribing. So these CTs are slowly conducting unmyelinated nerve fibres. We say they're exclusively functional in hairy skin, although 1 or 2 have been found in glabrous skin. Gentle stroking, they're also beautifully temperature tuned. So these nerve fibres respond optimally to a skin temperature touch, which is at skin temperature. So let's now look at a couple of possibilities as to what they're regulating. This is a study that I did with a colleague of mine in Valparaiso in Chile who had rodent models of stress. And this is a straightforward story here, is that the everyday rat was lifted from its cage and either stroked at 30 centimetres per second, which is a velocity that the CLTMRs won't respond to. Another group were lifted out of their cages and they stroked it 5 centimetres per second. And there's a couple of controls. During that 2-week period, for 3 or 4 minutes every day, all they had was fast touch or slow touch, but they were subjected to a chronic, unpredictable, mild stress paradigm, to the smell of cats, loud noises, you know, busy, horrible 2 weeks. At the end of that 2 weeks, we got the fast and the slow touch rats, and we found the most extraordinary response in terms of, just look at the paper is probably the best way, but basically if you throw a rat into a bucket of water, it won't, a depressed rat won't try and get out, it'll just roll over. But we looked at the response to that stressor in terms of corticosterone, and we found that those rats that had that slow touch had the resilience to stress in exactly the same way as a rat that wasn't stressed at all. Those that had the fast touch basically were not resilient to stress and were highly stressed. And we looked at a couple of other measures behaviourally, and again we see exactly what's happening here is that those rats that all they had was a few minutes every day of stroking touch were just resilient to all of these behavioural studies in terms of open field, and various other aspects of behaviour. This is a study that was done by a colleague of mine in Russia where we looked at children who were family reared. We looked at cortisol levels in children who were family reared at baseline, and they were within the normal range. We looked at those in institutions and those in foster care, measured their cortisol levels. We then put them through a 2-week paradigm where they basically had this, well, I call it this dose of vitamin T. They had 3 or 4 minutes every day of touch. That's all, no therapist, nothing else going on, just touch. We then measured their cortisol levels after that, and they dropped down to normal. Yeah, and this is quite remarkable. The only intervention that's caused this this reduction in cortisol is a tactile intervention every day. We've also, my colleagues in Brussels, have also, and Louise is one of these colleagues, who did this work looking at recording from these babies and looking at various measures of autonomic arousal, particularly this thing called respiratory sinus arrhythmia. And the only reason, by the way, that we get this kind of data is that Martine and Louise will only find the kind of responses to these stroking touch interventions in the baby's home with the mother. If you take these kids into a baby lab, you won't see this kind of behaviour. It's important when you're looking at social touch that the context is very important. And the other thing is just pain. And this, again, is another observation. I came across a group in Oxford who developed an event-related potential measured from the scalp of a baby, because you can't ask a baby whether it's in pain or not, but you can record a nociceptive response from the scalp. This is Rebecca Slater's group. And the bottom line here is that if you stroke the baby's leg at 3 centimetres per second before the heel lance, then that ERP basically disappears. So you've basically switched off and evoked a nociceptive response in that baby's brain by stroking it 3 centimetres per second. If you stroke fast, you do not get that response. So what do we know? We know that these nerve fibres are playing a fundamental role in a whole row of behavioural processes. And they have a stabilisation. They're calming you down, basically. Just one final comment. This loneliness study that was done by John Cacioppo, the odds ratio of a death from loneliness is 45%. But what nobody's looking at is that lonely people don't get touched. Yeah? They don't get their dose of vitamin T. So effective touch and development in preterm infants is where I'm now focusing most of my research, because these little preterm infants are covered in these little lanugo hairs. I'm not sure, maybe somebody will work this out, but whether these hairs actually are coupled into the C-tactile afferents as an encoding mechanism, whereby this is the skin telling the brain that the body exists through this process of amniotic fluid and touch. So we took this idea and did it in a study in Milan in a neonatal intensive care unit. We basically just stroked these babies at the preferred velocity, and we got a whole range of positive effects physiologically from just gently stroking those babies rather than touching them. So I took this idea and where my knockout model for how important the C-tactile is in the developing brain is when a preterm infant is removed from the womb and placed in the incubator, what doesn't it get? It doesn't get the touch it was getting in that third trimester. So that link between skin, brain and body is not happening. So we developed a little device with my colleagues here, including ADAS, where we're basically going to pop this mattress, basically will stroke that infant at the preferred velocity of CTs, and we'll look at a whole range of epigenetic and developmental markers that the babies that get this mattress will develop for more normally, because something like 20% of preterm babies develop autism. Many of them have some neurodevelopmental consequences for being born preterm. My hypothesis is that it's touch. Just a couple more slides now, another one I came across by a guy called Brian Gold in Lethbridge. He had an Alzheimer's disease model of mice. All he was doing was stroking these mice every day for a few minutes, and he found behavioural increases and also reduction in plaque formation. He also found by stroking these mice, that he could actually recover function after a brain lesion. And all again he's doing is touching, activating the CLTMR. And the post I had yesterday shows also that stroking touch improves the gut biome. So, two types of touch, discriminative are effective. I don't think they would operate separately. I think they must operate in some harmonious way in terms of the interdependence between the two. And the reason I say that is that we have this wonderful patient, Ian Waterman, who's lost all his myelinated nerve fibres. All he has left is C fibres. So he responds to itch, he responds to pain, and he'll respond to gentle touch, but only at a very minimal level. Yeah? He's not sort of screaming. So I think the fast touch stuff needs to be there as well. And again, these C fibres, I've already discussed, we have 3 of these societies. We're having our annual meeting in Jena in a couple of weeks for that C fibre. And just finally, there's audio here, or there should be.
Oh,
Can you hear that? Is that running?
Pops?
Hello, love. Do you have to do that?
Oh, can't a daughter give her old dad a hug?
I'm not a huggy person.
Oh, you're all stiff and knotted up.
That's normal for me.
You've got too much stress in your life, Dad. You know what's good for relieving stress?
Valium.
A big hug.
Hugging is a continental affectation, like cheek kissing and croissants. In England, we shake hands. A manly handshake is just as good as a hug, and it stops us catching each other's germs.
Well, I don't care. I'm going to keep on hugging you whether you like it or not.
So, thank you so much. That last clip, by the way, is not everybody particularly, craves a hug, so we need to be respectful that, we don't all want to be.
Thank you very much, Francis. Questions? Yeah, if you just wait for the microphone.
I've always found it quite interesting that you don't have, or you have a very low level of CLTMRs in glabrous skin versus hairy skin, because that presumably means if you're the stroker, if you're getting stroked, then your CLT or CT fibers are getting activated. If you're the stroker, that's not really happening. But I think if I have a stressful day at work, one of the first things I do when I get home is stroke my pets. And it obviously de-stresses me, and that's well known as well. So I assume that's a different mechanism there.
I think that's
not
learned than a secondary reinforcer. But you're absolutely right. I mean, during COVID, the brain knew it was deficient on this vitamin T. It didn't know about the CT. So in England, they all bought these bloody dogs, yeah? And why did they buy these dogs? Because they needed, as you already worked out, something to touch. But certainly the giver of touch has to also be rewarded as well as the receiver in evolutionary time. So the giver is also getting lower cortisol levels. They get the same buzz, but through a different mechanism, I think. Otherwise, it wouldn't have worked with the reciprocal grooming you know, you scratch my back, I'll scratch yours. You need to have that reciprocity. They're not here, the bottom.
Francis, you've shown the benefits of this type of touch in long term. We don't know much about the central circuits activated by CTs or CLTMRs, but do you think that we could find a potential therapeutic node that we could target to reduce the consequences of an absence of early touch life?
You mean through some kind of robotic stimulator or some way of...
Just some sort of manipulating activity within a particular
Well, that's
neuronal node or central circuit.
Baby mattress was tapping into that. I don't think the neurofiber, I'm going to say, gives a damn about who's stimulating it. So any device that's basically activated. This is why we built the robot, because we knew that a human giving that touch would add another dimension to it.
But I'm thinking more downstream of the afferent itself.
If we could,
So if we have things like conditions like neuropathic pain, chronic pain, whatever it is, people will target central pathways and manipulate activity there.
That is the big, we don't have a ligand, yeah? If we had the capsaicin we have for the nociceptor for the CT, we'd be off, because we'd be able to, because these nerve fibers are all in the epidermis, they're all out there. They should be an interesting, that RNA-seq paper that Hokan's group and Saad has published should give us some indication that we have a ligand now for this nerve fiber in human DRG. Is that right, Saad? So we're getting close to having a molecule. The thing about capsaicin is it activates the VR1 receptors on nociceptors, which is why it gives the burning pain. The same with menthol, activating the cool receptors, giving the sense. If we want that molecule for the CTs, and cosmetic companies will be over the moon, because every cream you're putting on is basically activating CTs as well as the process of actually touching. So grooming behaviour is more about feeling good than looking good, I think.
Yep. One final question here.
Do you think these fibres know the difference between this sort of pleasant touch and ticklish touch? Almost, because they're quite similar, surely.
That was Zotterman's first conclusion when he first discovered that C fibre that responded to slow touch. He thought it was a tickle. But they're too fast for the CTs. And this information is getting into the brain seconds after the stimulus, So I think the CTs regulate stress. I think that's what they're doing. We've oversold the pleasure bit a bit, because it's not that pleasant, in terms of sexual behaviour, for example. It's kind of lifts your mood a bit. So I think these things are regulating stress, as we saw with that Russian study, in terms of stress. And the rats, just a few minutes a day of that, and they were resilient to stress. And this is how critical this is, because the more we lose touch, the worse our mental health's going to get, because we're not getting this vitamin T top-up every day from the usual sort of behaviour that we've taken for granted. Adash.
I think that's also a really good example of the interaction between the A-betas and C fibers, because the force makes a difference, and very light forces will possibly activate CTs, but will activate a different type of A-beta fiber, whereas gentle, the kind of CT optimal touch, does tend to have sort of preferred force as well.
This is a hot topic. This microneurography, I'd say with the Saad and Adash are two people that can record from the CT. I say there's 20 people globally who can actually tap into that nerve fibre. It's incredibly difficult. You need a particular kind of personality. You can sit there for hours plugging away trying to catch one of these fish. But it is a first-order neuron. It's a beautiful technique, because you can't argue with it. It's just got everything you need in terms of its biological properties to stroking touch.
I'm conscious of time, but what I'm going to suggest is that I see there's some other hands for questions. If we can stay on at the end of the session, because the room isn't required, I think there might be an opportunity for some further questions. But I think we should probably move on, and I'm going to hand over to Miriam now, who's going to chair the next two or the last two presentations. So, Miriam.
So our next speaker is Dr. Saad Nagy from Sweden. I'm not gonna go for trying to pronounce the name of your university. Maybe you can you can do that. And he's gonna now, I guess, move a little bit away from the C fibers that we have now talked a lot about, and move towards E beta and the physiological and molecular characterization of human A beta nociceptors at baseline and following inflammation. Thank you.
Yeah, thank you, Mariam, and thank you to Journal of Physiology and Warren Macefield for helping organize this. Very briefly, the classical view is that touch is fast and pain is slow. So touch is signaled by thickly myelinated, very fast conducting A beta neurons, while pain, temperature, and as Francis just showed, certain aspects of touch are signaled by thinly myelinated or unmyelinated A delta and C fibers. So the dichotomy is you have fast touch, slow pain, and that's what typically the neurophysiological testing is based on. But if we look at non-human mammals, we do find that there is a prevalence that A beta range pain sensing neurons, or nociceptors as they're called, do exist, the prevalence of which varies across species. So we, some years ago, we asked a very simple question. Why do pain signals not travel as fast as touch signals? And to do that, we, as Miriam and Francis described, we use the technique of microneurography. We do it under ultrasound guided, under real-time ultrasound guidance. So we insert a very, very tiny microelectrode. The shaft is about 200 micrometers, the tip maybe 5-10 micrometers, into the skin, and under real-time ultrasound guidance, we make small adjustments to the electrode until we can impale a nerve. This is in an awake human. It can be any nerve, trigeminal or peripheral. We've mostly worked with peripheral nerves now. And here's an example of the ultrasound image. So this is a recording from the upper limb, where this is the humerus bone of the upper arm, and here is our microelectrode approaching the radial nerve. And when we get into the nerve, we make we mechanically. So here, our approach is somewhat deviates from what Miriam described. So instead of electrical, we look for single afferents using mechanical stimulation. So we apply a range of mechanical stimuli, brushing, pinching, poking, so on, until we, and this is within the innervation zone or the fascicular zone of the nerve fascicle, until we can identify a single axon, a single afferent. At which point, what we see during a recording in the lab is something like this, where each vertical line represents an individual action potential from that recorded afferent. And we know it is from the same afferent as, since it's restricted to that site of skin that we stimulate. So only when we stimulate that part of skin would that elicit a response. And then offline, we look for spike morphology and so on to confirm that it is indeed the same spike. So at that point, we then switch to more well-controlled stimuli and look at the tuning properties and so on. So typically what we find in these sort of recordings are two big groups. We find the neurons that respond to gentle touch, and we find neurons that don't. Those that respond to gentle touch typically have low mechanical thresholds. Those that do not respond to gentle touch have high mechanical thresholds. What got us quite excited was when we measured the conduction velocities, we found that within the high threshold range, there were neurons that were signaling just as fast as the A beta touch neurons. We, of course, did record from the classical C fibers, which are very, very slow. But this was quite striking, and that the high threshold brush insensitive neurons in the A beta range. And we recorded from two nerves, peripheral, perineal and radial, and what's well known in neurophysiology, the conduction velocities are somewhat faster in the upper limb, and that's what we see in our single unit recordings as well. So to define a nociceptor or pain sensing neurons, there are two particular features. It's high threshold, so it will not respond to gentle touch. We don't want our pain-sensing neurons to respond to gentle touch, and unless it's chronic pain, of course, then they do, and the capacity to encode noxious forces or tissue-damaging forces. So we looked at how well their tuning properties are, and across a number of different standard neural metrics. This is based on the pattern of action potential firing. We find that they do encode noxious forces very well. In other words, the stronger the poke, the bigger the response. And since these are human participants, we can ask them what they feel, and we find, well, predictably, you poke harder, it's more painful. And so when you compare the psychoneural data, you find a very neat correlation between the two. So we think these are high threshold, very fast conducting primary afferents in human skin, quote, painful indentation. So these are A-beta nociceptors. Then another thing you can do with microneurography is, since you're using an electrode, and while you're recording from an electrode, you can also send brief electrical pulses. And when you do that, and it's been shown over several decades, and you nearly selectively activate an A-beta touch fiber, the percept can vary anything from pressure to tapping, flutter, vibration, but it's never pain. And these are very, very low currents. I mean, 5-6 microamps. And when you do the exact same stimulus intensity, when you deliver the same intensity to these A-beta nociceptors, it's described as sharp pricking pain. So this is all published, and I'll now switch to more unpublished work. But the summary here is A-beta nociceptors, conduction velocities similar to A-beta touch neurons, high threshold, brush insensitive, can encode noxious skin indentations, and when selectively activated, produce painful percepts. So we then asked, well, do they respond to temperature? I mean, certainly within the C fiber domain, we find a lot of polymodality. So do they respond to temperature? And we delivered temperature at the physiologically mapped out receptor field, and we find that a subset of them do. So not all of them, but some of them do, and I'll discuss that in more detail. And those that respond to cooling, they respond not only in the dynamic phase of cooling, but also in the sustained phase of cooling, whereas they do not respond to heating. So then we wanted to know what's the molecular identity of these, and we reached out to a group at UPenn who were doing single-cell RNA sequencing of human DRG neurons, and within their sequencing data, they had identified 16 clusters of human sensory neurons, and there was one particular cluster that caught our interest. It's called the H, or for human, PEP-KID. So it's a peptidergic type of neuron that expresses a receptor tyrosine kinase. And the reason why it caught our interest was there were a lot of similarities between what the transcriptomic data had revealed and what we were recording from these A-beta nociceptors. For instance, there was NEF-H expression, and indeed we find that these are myelinated in our conduction velocity data. There was no TRPV1 expression, and we don't find any heat responsiveness. Interestingly, there was TRPM8 expression, and while we had shown that a subset of A-beta cool positive nociceptors respond to non-painful cooling, we wanted to test whether that response is dependent on TRPM8. So to do that, we went back to microneurography. We recorded from an A-beta cool positive nociceptor. So it's very typical. They don't respond to soft brush, but if you use a very rough or coarse brush, they will respond. Then we tested its cooling response, and yes, it did respond. At that point, we injected high-dose menthol around the injection site as a means to desensitize the TRPM8 channel. And when the cold perception was lost, we applied mechanical stimuli to the receptor field, and the neuron was still there. It was responding mechanically, so the recording was still intact, at which point we tested cooling, and there was no response. So that to us confirmed that these A-beta cool positive nociceptors rely on TRPM8 for their cooling response. So that confirms that. And then is the question about piezo2. Now, Alex Chester's lab have worked over the past few years on patients who have a congenital mutation in piezo2, so, and they have severe touch and proprioceptive deficits. But because of those deficits, it is not feasible to do microneurography. However, we did do some psychophysical testing in those patients. And what we and others have shown before is that pain is typically normal in patients who have no piezo2. And this has been tested for pinprick, for pressure pain, and pinch. Interestingly, there's only one type of pain that they don't feel, and it's pain to hair pulling. So they can feel poking pain, but they don't feel pulling pain. So there's a very clear deficit in pulling pain. So we thought, well, our A-beta cool positive nociceptors, do they respond to hair pull then? And yes, they do. They respond very vigorously to hair pulling. And then we tested a range of pulling forces from non-painful to painful, and we find that the response is beautifully tuned to the intensity of hair pulling. So this is single hair pull. Yeah, so across a range of forces until the hair is eventually out. They respond beautifully. The stronger the pull, the greater the response. And that corresponds very nicely with psychophysical data for pain intensity to hair pulling. We collected this data from all afferent types. Just in the interest of time, I'll only show comparison data from C nociceptors, and there you see that the response plateaus out. In fact, it starts to, C nociceptors tend to drop out as we approach very high pain. So you don't find a strong concordance there. So we think the HPEP kit transcriptomic cluster is what in our physiological recordings we call the A-beta cool positive nociceptors. So the key points there, well, in the interest of time, I didn't show data, but we also did immunohistochemistry of human skin biopsies, and at least we find that there were myelinated nociceptive endings around human hair follicles, and they found these circumferential endings. So there is an anatomical association there. So we think they're associated with human hair follicles. They're cooling and menthol responsive, and that cooling response is dependent on TRPM8. They encode acute hair pulling pain, which we believe is dependent on piezo2. And this is the first demonstration of the role of piezo2 in an acute pain sensation. And we confirmed that the transcriptomic class is the HPEP kit. So just to summarize this part, there are at least two classes of A-beta nociceptors. The A-beta cool positive nociceptors, we mapped them to a specific transcriptomic class. They express piezo2 and TRPM8. And then there is the other class which do not respond to cooling. And they do not respond to hair pull either. We think the tentative molecular identity is another transcriptomic class called the HPEP NTRK3, but this is work in progress. So the next question was, okay, well, they do exist. There are likely two types, but are they relevant for pain hypersensitivity? And the temptation was to switch to pain pathologies, but we thought we should start with an experimental model first, where we have control of the different stimulus parameters. And so we switched to, we recorded from these A-beta nociceptors in an acute heat-induced inflammation model. So essentially what we do is we record from an A-beta nociceptor. When we have characterized the unit and have a stable recording, we then deliver heating for 2 minutes. So it's a modified burn model, but does not cause any permanent injury. It's in fact a mild model of inducing inflammation. And what we find is that after 2 minutes of heating on the receptor field of A-beta nociceptors, there was a striking drop in their mechanical thresholds, as you can see here. So these are the mechanical thresholds at baseline, and then these are the mechanical thresholds after 2 minutes of noxious heating at the receptor field. And this seems specific to A-beta nociceptors. We recorded from all other afferent types we can find. For instance, the C nociceptors, we don't see, we don't find them sensitized. Likewise, with the A-beta touch, we don't. So not in this model anyway. Yeah. And what's quite interesting is that if you break it down into the two types of A-beta nociceptors, then the A-beta cool negative nociceptors, the mechanical thresholds in them drop to such an extent that they actually become brush responsive. So here are the mechanical thresholds of A-beta cool negative nociceptors. And these are recordings from 13 units. And in 10 out of 13, the mechanical threshold dropped to a tactile range, which in our hands is typically at or below 1.6 millinewtons. And then they become brush responsive. So we think this could potentially be a substrate for tactile allodynia. And the A-beta cool positive nociceptors, what's interesting about them is while the mechanical thresholds don't drop to the extent that they would become brush responsive, and hence we don't see a brush response in them, but when we apply cooling, we find that there is a striking amplification of their cooling response. So here is data for cooling at baseline across a range of temperatures. So it drops from a baseline of 30 down to 0. And then here is data to those same stimuli after 2 minutes of noxious heating to the skin. So you can clearly see that there is a much greater response now. We then looked at, well, how well do they encode mechanical stimuli before and after inflammation? We know as nociceptors, they encode mechanical stimuli very well, well into the painful range. And here what you see is data, paired data comparing baseline and inflammation responses. So each circle here represents a single A-beta nociceptor, and it's color-coded, so it's comparing baseline to inflammation, and you can see across all neurons, all A-beta nociceptors we recorded, there's a clear amplification of response after inflammation across a range of indentation forces. And when we compare that to psychophysics, we again find a good concordance between what we see in our neural recordings and what the subjects report in psychophysical testing, showing increased mechanical pain. Now, this is something that I find very interesting. So what we find is that, so during these experiments, we can physiologically map out the receptive field. So we know exactly where the receptive field is, what's the architecture of the receptive field. But what happens after 2 minutes of heating is that there is a remarkable expansion of the receptive field. So this is still work in progress. It's early data. But here, what you see are receptive field changes for A-beta nociceptors, C nociceptors, and A-beta touch fibers. And while the C nociceptors and A-beta touch fibers do not change, you find this remarkable expansion of the receptive field. So this is at the level of a single order neuron. It's a primary afferent. And it's quite an expansion. So this was the receptive field. So here's an example of that from one recording. This was the receptive field at baseline, and this is the expansion of the receptive field following inflammation. So we think this is quite interesting, and it probably means that, you know, following an injury, that whole the entire neighboring area, the region around the injury is hypersensitive, so as to protect that site of injury and to facilitate healing. Right? But this is quite remarkable. I think it's. I grew up in, well, I had my training in a lab which was very label-lined, and it's I find that quite amazing that just a mild inflammation stimulus can completely break the label, just change the labeling. You know. And psychophysically, I mean, we do, again, there's a good concordance. We do find that following our inflammation, in our inflammation model, there is expansion of the area of hyperalgesia or pain hypersensitivity. So to summarize, both A-beta cool positive nociceptors, which is the H-pepkit class, and A-beta cool negative nociceptors become sensitized after heat-induced inflammation, where we find a drop in their mechanical thresholds to the extent that they become brush responsive. We find enhanced responses to noxious mechanical stimuli and expansion of their receptive fields. Our preliminary psychophysical data shows that there is increased mechanical pain sensitivity and an expanded area of hyperalgesia. Between the two subtypes, the A-beta cool negative nociceptors, they develop a novel brush sensitivity, while A-beta cool positive nociceptors, they show an amplified response to non-painful cooling. In this model, at least, the C nociceptors and the A-beta touch neurons show no change in responsiveness, indicating that the effect is specific. So this demonstrates that A-beta nociceptors undergo rapid gain of function changes following acute inflammation. And in our ongoing work, we are now looking into the mechanisms of this sensitization. So with that, I'll thank all the collaborators and the organizers again, and thank you. Happy to take questions.
Yeah, thank you very much for this great talk. We've established also a lot of new unpublished data. So are there any questions from the audience for our speaker? Yeah. I think they're coming with the microphone.
Thank you, lovely talk. You did touch upon one aspect that I was meaning to ask about with regards to the type of peripheral endings that these A-beta nociceptors have, and you did mention that you find these types of endings from myelinated afferents associated with hair follicles, but hair follicles, depending on the type of hair, can have different types of specialized endings. And when we talk about nociceptors or unmyelinated C fibers in particular, apart from the CT afferents, they tend to have free nerve endings according to textbooks. So where would you actually sit with, and I know it's difficult to correlate your microneurography recordings with histological preparations, so where would you hedge your bets? Do these types of afferents have specialized endings that are associated with hair follicles, or are they free nerve endings?
Yeah, so I think we think at least the TRPM8 expressing A-beta nociceptors, they have specialized endings. And we think they form circumferential endings around hair follicles. We know less about the other type. It could be free nerve endings, but we know less. It's just that for the pure mechanonociceptors in the A-beta range, we haven't quite figured out their molecular identity yet, and so it makes it harder then to. But at least for the cool positive, which we think are the PEP-KIT class, we think they form these circumferential endings around human hair follicles. And we've been collecting human skin biopsies and running immunohistochemistry, for instance.
More questions? Yeah, there is another question in the front.
Great talk, thanks. It's just to follow on from David. So you show this innervation of hairy skin with the A-beta nociceptors. Do they target glabrous skin? Do you know?
We haven't recorded from glabrous skin yet, so we don't know. We have indirect data. So in parallel studies, we have been looking at the nociceptive withdrawal reflex. Because I mean, if you think about fast conduction, I mean, intuitively, it's the fast withdrawal where the conduction speed would be critical. And there we do, I didn't mention that in today's talk, but there we do find a role of A-beta nociceptors, and or what we think are A-beta range pain signaling neurons, and that's data in glabrous skin. So we have indirect evidence, but we haven't actually done microneurography in glabrous skin.
I have recorded from one A-beta high threshold mechanoreceptor in glabrous skin in Warren, in fact.
Fantastic. Well, there you go. They do it.
Is there any selective loss of these peripheral nerves in things like diabetic neuropathy? Or we know the C fibers go, and paradoxically they get burning, intense pain. Do they survive some kind of neuropathic loss?
So those are the sort of questions we're starting to look at now. I mean, the temptation was, after having characterized them, the temptation was to go straight into clinical pathologies. Mm-hmm. But the challenge there was that if and as we clearly see now, that these A-beta nociceptors undergo very rapid gain of function changes, then how would we classify them? Because if an A-beta nociceptor is now responding to brushing, then with our experimental model, we record from them first and then induce inflammation so we can track changes in real time. So whatever those plastic changes are, we are following them in real time. But in clinical pathologies, those changes may have already happened. So that was the reason why we decided to work with an experimental model first. But now we are running experiments in fibromyalgia and migraine, for instance.
Okay, so maybe the one last question, so because then we can also have a look at the time.
The rapid expansion of the receptive field for the A beta nociceptors, that was interesting, because that presumably means that they've got a very wide area of peripheral endings, and under normal circumstances a lot of that is shut down. So do you have any evidence for that? Do you know anything about that?
No, we don't. We're still looking into that. I mean, Martin Schmelz showed that for silent nociceptors back in the 90s. So it's quite interesting. So what we think that there are, it's pure speculation. We think that there are silent areas in the receptor field. And one possible explanation could be that following sensitization, when there is this drop in mechanical threshold, then stimuli which were previously subthreshold can now elicit a response. But, I mean, we don't quite know what is the mechanism of sensitization. So I think that we need to, and of course, if anyone has any thoughts on that, I think it's critical to figure that out. I mean, is it phosphorylation of PSO2? What is going on? Whatever it is, it's quite rapid. It only takes 2 minutes. So I guess it's not enough it's too short a window for any elaborate gene expression changes. So what's happening? And I think that mechanism we need to understand better, and that may help address some of the other questions. Exactly, yeah. Exactly.
Okay. Yeah. So I guess also due to time's sake, we're going to move on to the next talk. Thank you very much. So our next speaker is Dr. Carol Torsney, also from Edinburgh, and her talk is titled Tactile and Pain Processing in a Neurodevelopmental Disorder rat model. Thank you.
So the work that I'm going to present today has been carried out by Katerina Mazur and Ying Zi, who are both here in the audience, in my lab in the Simons Initiative for the Developing Brain at the University of Edinburgh, where we have started to explore tactile and pain processing in neurodevelopmental rat models. So as the previous speakers have beautifully introduced, this capacity to detect and experience pain and touch is vital. It enables us to safely and richly experience the world. And there are multiple lines of evidence from both clinical and preclinical studies that this somatosensory system is altered in neurodevelopmental disorders. So to explore this, we have asked in neurodevelopmental disorders, is there disrupted spinal somatosensory processing? And to explain why this is a relevant question to ask, in the adult spinal cord, for the most part, pain and tactile inputs are anatomically segregated, with pain inputs largely targeting superficial regions and tactile inputs targeting deeper layers. And local and descending control is mature. Whereas in the newborn, initially those tactile inputs are much more widespread and overlap with the pain inputs, and those local and descending controls are immature. And in the newborn, this is associated with a tactile hyperreactivity that's evident in spinal reflex responses. So in the newborn, spinal reflex tactile hyperreactivity, and in the adult, a tactile normal reactivity. So if you like, spinal reflexes provide a window into the maturity of this system. So the postnatal development of this system is activity dependent, and given that a number of genes associated with or mutated in neurodevelopmental conditions are key players in these kinds of activity dependent processes, we hypothesised that this process may be disrupted in neurodevelopmental disorders, and that may be evident as altered spinal reflex responses in the adult. So to explore this behaviourally, we've assessed spinal reflex responses to a range of noxious stimuli, and for tactile, we've looked at reflex responses to tape application, paintbrush stroking, and we've also looked at fine sensory motor control using a sunflower seed manipulation assay. So we've looked at this in the context of SYNGAP1 haploinsufficiency. And this is a neurodevelopmental disorder associated with intellectual disability, epilepsy, and autism. And it's caused by mutations in the SYNGAP1 gene, and this leads to a reduced SYNGAP protein production. Oops. So individuals with this condition have altered sensory responses, altered pain responses have been reported, as well as tactile seeking and tactile aversive behaviours. So to explore this, we've studied a SYNGAP heterozygous rat model that has 50% of the wild type protein expression, mimicking the clinical condition. So SYNGAP is a RAS-RAP GTPase activating protein that's expressed in neurons where it has multiple functions. It's involved in neurite outgrowth, and it also regulates synaptic function. So we were interested as to whether loss of SYNGAP has any impact on touch and pain processing in this rat model. So we'll start off looking at noxious mechanical stimuli, here applied to the hindpaw. So I should say all of the data is separated by sex, and we have our wild types in white and the hets in blue throughout. So basically the mechanical force required to drive a reflex response is unaltered between genotypes, and the time taken to withdraw from a suprathreshold mechanical stimulus is also unaltered, and the response score to pinprick also unaltered. So we have an unaltered noxious mechanical reactivity. If we look at noxious thermal stimuli, and in our Hargreaves assay, the time taken to withdraw from a noxious heat stimulus is unaltered between genotypes, although we've got a sex-dependent effect, and similarly, an unaltered withdrawal latency to noxious cold. So unaltered noxious thermal reactivity. So now if we move to tactile stimuli, and we're going to start off with glabrous skin. Here we use this dynamic paintbrush assay where we have a scoring system to quantify the level of reactivity. And we see a genotype effect with a reduced tactile reactivity in the heterozygous rat model. Sticking with glabrous skin, we also had this sunflower seed manipulation assay, which involves tactile processing in the glabrous forepaws. And we see an interesting phenotype here. While most of the wild types eat and deshell 10 seeds, the heterozygous model, they deshell far fewer seeds. And if we look at the time taken per seed, that is increased in the hets. Now, clearly in this task, there are many different factors that may feed into this behaviour and phenotype, but given we see a tactile deficit in the paintbrush assay, it could be that a tactile deficit feeds into this phenotype. So certainly from the paintbrush assay, we have a tactile hyporeactivity. So moving to hairy skin, here we have our back tape assay. So here a piece of electrical tape is placed on the back of the subject from the nape of the neck, and this induces a wet dog shake response, which is a reflex response involving a spinal parabrachial circuit. And I'll just let you see it, it's very cute.
In this assay, we see a reduced number of wet dog shake responses in our HEP model. And interestingly, overall, females show more of these wet dog shake responses. So again, in hairy skin, tactile hyporeactivity. So we've also conducted a variation of this test where a bit of tape is placed onto the hairy surface of the hindpaw, here a little bit of masking tape, so that the animal can remove the tape if they want to. And you can see that here. So in this particular assay, again, we see an interesting phenotype. While most of the wild types remove the tape, as I think this one is just about to, the hets are much less likely to remove the tape. Again, clearly, from having seen the video, it could involve some sort of motor or coordination deficit. But interestingly, if you look at the time taken to first respond to the tape, and that can just be sniffing at or licking at the tape, or reach a cutoff, the hets take longer to respond, which could be consistent with a tactile deficit. Also, the percentage of time spent responding to the tape when it's attached is also reduced in the hets. So potentially further evidence for a tactile hyporeactivity. So we've got various lines of evidence for a tactile hyporeactivity. So is there disrupted spinal somatosensory processing? So importantly, with Westerns, we've shown that SYNGAP is indeed expressed in the dorsal horn of the spinal cord where those pain and tactile inputs are processed, and is reduced by 50% in the HEP model. So to explore mechanism, we first looked to see if there was any alteration to the anatomical wiring of touch and pain inputs to the spinal cord. So as I highlighted earlier, initially those tactile inputs are more widespread, and then they gradually withdraw to the adult deeper location. So we wanted to check if this process had occurred in our adult tissue. So to look at this, we use the pain marker IB4. And basically, if you look at the area of this staining, the thickness of that band and intensity is unaltered between genotypes. And if we use VGLUT1 as a marker of the tactile inputs, here, if we measure the depth, if you like, to the start of this staining and the area above the staining, that's unaltered between genotypes, suggesting that that withdrawal has taken place normally. So overall, certainly at a gross level, there doesn't appear to be any change to the termination pattern of pain and tactile inputs. So given we have this tactile phenotype, although we predict a spinal mechanism, it was important to check the tactile innervation of the skin, which we've done here for a subset of the tactile afferents. We've looked at the innervation by tactile corpuscles. And so here, both the density of these kind of dermal papillae structures and their innervation with tactile corpuscles is unaltered between genotypes. So given we don't see any obvious change to the anatomical wiring, we then asked, is there any functional differences at the level of the spinal cord? So as I said, SYNGAP's expressed in neurons, where it's highly concentrated in the postsynaptic density of glutamatergic synapses, where it's thought to regulate AMPA receptor anchoring. So we asked, is there disrupted glutamatergic synaptic inputs to tactile processing neurons in the HEP model? And to look at this, we've moved to patch clamp recording and spinal cord slices, here where the sensory nerve is left attached, so you can electrically stimulate those incoming tactile and pain inputs. And we can make recordings from neurons in this superficial pain zone or in the deeper tactile zone. And by recording under these conditions, we can record these excitatory postsynaptic currents driven by direct tactile and pain inputs. Now, these experiments are carried out in tissue from about 3-week-old animals, and so it was important to check that our phenotype is present at this age, as all the behaviour I've shown you has been from adult subjects. And indeed, in that backtape assay, we see a reduced number of wet dog shakes in the HEP model. But before going on to our spinal recordings, it was really important to check that the excitability of the sensory input itself was not altered by genotype. So to look at this, we conducted extracellular population recordings from isolated sensory nerves. And in these compound action potential recordings, as you gradually dial up the stimulation intensity, you recruit the different classes of sensory input. So at low intensities are fast-conducting A-beta fibres. Then as you dial it up, you also recruit the medium-conducting A-delta inputs. And lastly, at the highest intensities, you also recruit the fast-conducting C-fibre component. And essentially, the threshold, amplitude, and conduction velocity of all of these components is not altered by genotype. And given we have a tactile phenotype, we also checked the A-beta refractory period, and again, that is unaltered by genotype. So the excitability of the sensory input itself is unaltered in this model. So we can now move to our patch clamp recording in slices. And in terms of characterising the input to these neurons, we can use our knowledge of threshold and conduction velocity from the last slide to classify inputs. And we can use high-frequency stimulation to tell whether inputs are monosynaptic, direct, or polysynaptic, indirect. And that's just shown here. This response has 2 clear components. And if we stimulate at high frequency, this one is reliably evoked with little change in latency, indicating monosynaptic, whereas this response fails, so it's polysynaptic. So we can use this to look at direct sensory inputs onto spinal neurons. So if we look first in the tactile zone, here we have an example of an excitatory postsynaptic current in our wild-type subjects versus a het, and you can see that the peak amplitude of that response is reduced, and that is quantified here. Whereas if we look in the pain zone, we don't see any difference in the peak amplitude of the response. So this indicates a potential spinal deficit in the processing of tactile rather than pain sensory inputs. And we've also looked at this in spinal reflex network recording, which you can conduct in this hemisected spinal cord prep, where you can electrically stimulate the incoming sensory input and record the motor output of the spinal cord in the ventral root. So here you're just looking at the activity in the neural circuit that underlies a spinal reflex response. And you can see here in the wild type example, we first see a response at 7.5 microamps, and whereas in the het example, we don't see a response until higher intensities. And that's just shown here. We have an increased threshold in the hets, but the maximal response is unaltered. And if we look at the gradual recruitment of this response as you dial up stimulation intensity, the intensity required to recruit 25% of the response is increased in the hets. So given that the threshold for driving a response and recruiting 25% is very much in the low threshold tactile range, again, this is evidence supporting a spinal deficit in the processing of tactile rather than pain sensory inputs. So to summarise, the SYNGAP het rats are hyporeactive to tactile but not noxious stimuli. It appears that the gross, spinal and skin sensory innervation that we've looked at does not appear to be altered, and our functional data would suggest a spinal deficit in the processing of tactile sensory inputs. And lastly, I just want to highlight a couple of things. We have another poster tomorrow on a slightly different topic, post-surgical tactile pain, and there's a flash talk for that at midday. And we're also hiring, got a short-term postdoc for a post-surgical pain project, so if you're interested, please get in touch. Lastly, big thanks to Z and Kath, who conducted the work I presented today. Peter Kind, who's the director of the Simons Initiative for the Developing Brain and Lab for Support. Sally Till, who created the SYNGAP rat model, and the Simons Initiative for the Developing Brain for Support. Thank you.
So thank you so much, and I think we are starting a bit late. We are perfectly in time, 11:15, so there is still room for questions, and we already said we could have if there are follow-up questions for or discussions for the other talks, we can also have a little bit of that. So questions from the audience? So maybe I can start, and maybe then the others have also the courage to ask some more questions. So I'm not sure, maybe I missed it in the beginning, but do your findings from your rodent model really reflect also what you see or what clinicians see in patients, that kind of deficits they have? They also have, or do they also show deficits in pain sensation?
Yeah. So to touch on the kind of pain aspect, a lot of the clinical reports with respect to altered pain responses comes actually from parent or caregiver reports, as many of these individuals may be non-verbal or restricted communication. So given that those are reports such as that, it probably more reflects pain tolerance or response to injury, whereas what we've looked at here has very much been acute pain reflex responses. So we're also now looking at those other dimensions of pain as well.
Okay. So other questions from the audience? Yeah.
Have you investigated the difference in processing of static touch stimuli versus dynamic stimuli?
So I guess behaviourally, we've done the von Frey punctate, we've done the dynamic paintbrush on glabrous skin, which is probably an A-beta fibre stimulus there. But we are interested in exploring the more kind of effective touch aspects on hairy skin also. Yeah. But in terms of at a spinal level, we can differentiate a little bit between the types of sensory inputs in our EFIS recordings, but we just don't have the kind of numbers to make those kind of judgments at an EFIS level.
Anything about inhibition in the dorsal horn? Have you noticed any differences?
We haven't, but it's an interesting question to ask. There's obviously recent work that's shown in a neuroligin 2 model that there is reduced feed-forward inhibition at the level of the spinal cord in that model, but there's a tactile hyperreactivity, whereas here we've got the hyporeactivity.
Okay, any more? Yeah, questions?
Thank you, very interesting. You mentioned the wet dog shake, and that reminded me of a paper that just came out a few months ago, showing that it's the CLTMR, spinal parabrachial pathway.
It's really interesting, yeah.
I wonder what, whereas in your model you don't find any, you find a wet dog shake effect and yet no C fiber, or did I understand that?
No. So in the wet dog shake response, we see a reduced response, and that paper you refer to from the Ginty lab has shown that that wet dog shake response involves a spinal parabrachial circuit that is driven by CLTMRs. So what that actually implies that given we see a reduction in the wet dog shake response, it may indicate that there is a deficit in affective touch, and we need to look directly at that, and that's what we're currently doing in behavioural studies.
Just an interesting observation from that study I did with the Oxford group when we stroked at 3 centimetres per second and the ERP was basically disappeared. We also measured the EMG from the leg, and there was still a reflex pull back.
What was that in the young?
And I haven't worked that one out, but that's fascinating because clearly the circuit isn't fully mature, so they had the withdrawal reflex.
Yeah.
However, the pain ERP had basically disappeared, so there's some neurodevelopmental angle here that we need to look at.
Yeah, and one thing we're doing now is trying to look at this through development and look at that hyperreactive reflex over the course of postnatal development.
But they didn't usually think that preterm babies felt pain, of course, didn't they? Which is
another story.
Disaster, quite frankly. Baby Jeffrey is the case. But, they operated on him without any anaesthetic. Got that anyway.
I'm conscious of time, but I also sort of noted that there was, I think, a few questions for previous speakers that weren't answered. I don't know if there's anyone with a pressing question that want to ask it just now. I see the hands going up. If not, I'm sure the speakers will be hanging around the podium for a few minutes afterwards. Really, all that remains for me, I just want to thank our speakers again. So I want to thank Miriam, Francis, Saad and Carol for a really excellent symposium. It's not my area of research. Those of you who know me know it's not my area of research. But it was, I thoroughly enjoyed it. I learned a huge amount, and I can see huge potential opportunities once we understand mechanism and understand, gain insights for potential therapeutics as well. So I really find it fascinating. I'd like to thank Physiological Society for its sponsorship. I would like to remind you all of the special issue that is manuscript submitted by early October. I failed to say that a great opportunity to speak with Carol, because Carol, along with Vaughan and Rochelle, are coordinating that special issue. So if you want to speak to Carol about that, then Carol is around for the rest of the meeting as well. If you want to go to the FysSoc stand, plugging it again, Alex and Dariel are here as well. So all that remains for me to do is to thank everyone. Thank you also, colleagues from BNA, for operating the mic and the AV as well. So thank you very much.